Welcome to Learning Management System, Commissionerate of Collegiate Education, Vijayawada, Andhra Pradesh. Myself, Indravati, Assistant Professor in Biotechnology, KVR Government College for Women, Autonomous, Kurnool. Dear learners, the subject recombinant DNA technology is made easy for you. Do follow all my video lessons to cover different topics related to this subject. Dear learners, today's topic is Introduction to Restriction Enzymes, and you will be learning what is a restriction enzyme and the discovery and the biological significance of these enzymes, the mode of actions and the types of cuts produced by these restriction enzyme, and also at the end you will be learning about the applications of restriction enzymes. Let's learn about restriction enzymes in detail. First, coming on to the definition. So, restriction enzymes are endonucleases which cut the DNA sequence at specific sites known as restriction sites or recognition sites. So, these enzymes are simply called as molecular scissors. So, you know that scissors are used to cut the things. In the same manner, the restriction enzymes are nucleases which cut the nucleic acid, and these nucleases are also endonucleases, that is they produce internal cuts, that is within the 5' and 3' and they will be producing the cuts, and also these internal cuts are also highly specific, that is these enzymes they recognize certain sites in the genome and produce cuts within that site. So, these sites are called as restriction sites or recognition sites. For example, EcoR1, it is isolated from the bacteria E. coli, and this particular EcoR1 is having the ability to recognize this GATTC sequence. So, this is a 6 base pair sequence. So, wherever this sequence is present in the genome, that sequence is recognized and it is producing cuts in both the strands of the DNA, that is here I have represented with star mark. So, between G and A it is cutting by breaking the internal phosphodiester bond in the upper strand as well as the lower strand. So, in this manner you will be getting two fragments. Okay. So, in the whole you can define restriction enzymes as endonucleases which produces internal cuts at specific sites. Okay. Then coming on to the history. These enzymes were first isolated in the year 1970 and the enzyme was HIND2. So, a lot of work was done on these enzymes and 3 scientists, Nathans, Arbor and Smith, were awarded Nobel Prize in the year 1978. And coming to the biological significance. So, why these enzymes are present in nature? So, these enzymes are most prominently they are present in bacteria. So, what is the purpose of this presence? So, these enzymes they act as immune system. So, in our body, how the immune system works, it is having the ability to distinguish between self and foreign, and whenever a foreign agent comes, our body shows immune response and expels the foreign agent from our body. In the similar way, the bacteria also, whenever it is attacked by virus, so whenever it is attacked by a virus, the viral DNA it is injected into the bacteria it activates its restriction enzymes, and these restriction enzymes they cut the viral genome into pieces and it expels it from its body. So, you will be getting a doubt, that is the restriction site, for example the EcoR1 site, GATTC. GATTC may also be present in this bacterial genome. So, how it is able to protect its own DNA and how it is able to cut the foreign DNA means this bacteria along with the restriction enzymes, along with the cleavage property, it is having methylase property also. Due to this methylase property, the recognition sites of its own DNA, they are methylated. So let's see here. So that is this is the sequence GATTC. So if it is that is the self DNA is methylated. So when it is methylated means certain methyl groups are added to the nitrogen base. When the methylation is occurring, the enzyme is no more, it is able to recognize this site and no breakage is occurring. So whenever the methylation is not seen, that is it is seen in case of the viral DNA. So here no methylation, so this site is recognized by the enzyme and it is broken into pieces. So in this manner the restriction enzymes, they combine with the methylase function and it is having restriction and modification system. It is existing as a combination restriction and modification system. In bacteria it will be acting as immune system and it will be acting as a defense mechanism and it will be killing the virus DNA. So coming to the mode of action, so we are saying that the enzymes are producing internal cuts, so at also at specific sites where it is cutting means, suppose, and you know that this DNA is made up of this DNTPs, deoxyribose nucleotide triphosphate. These are the monomeric, each monomeric unit is joined by means of the other unit by means of a phosphodiester bond. This phosphodiester bond is hydrolyzed or broken by the by this particular enzyme. So the restriction enzyme is able to cut the DNA by hydrolyzing the phosphodiester bond present between this adjacent bases. So in this manner you will be these two fragments of DNA are formed, and these cuts are also produced in both the strands, double stranded DNA, both the strands they are cleaved. So in this picture we will be explaining how the restriction enzyme is binding to the DNA strand and how it is producing internal cuts. The restriction enzyme here taken is BAMH1. So first it shows non-specific binding. So when the recognition site is nearer to the region, then this particular enzyme, it shows sliding movement, and when the recognition site is far away, the enzyme will be showing hopping and jumping movement for searching its site. So by means of random binding or non-specific binding on the DNA, the restriction enzyme will be searching for its recognition site. So once the site is recognized, then it will be coupling and binding to this site. After that it will be showing its action, that is the breakage of the internal phosphodiester bond. So this BAMH1 is having the ability to produce cuts in the, that is staggered cuts between G and G in the upper strand and also in the lower strand. In this way these two fragments are generated by this internal cuts produced in the double stranded DNA. Next, coming to the type of cuts produced by the restriction enzyme, so staggered cuts and blunt cuts. So first let's see the blunt cuts. So if the enzyme is having the ability to cut the restriction site at the same position in both the strands, that is exactly in the middle, the type of cut is called as blunt cut, and it generates DNA fragments having fleshy ends. Okay, so here the breakage is at the same position in both the strands. If the breakage is 2 to 3 base pairs away from this middle portion, then it is called as staggered cut and it will be producing sticky ends. The sticky ends will be having a single stranded regions at either in 5' or 3' end. So here these single stranded regions are called as overhangs, 5' or 3' overhangs, and they are also called as cohesive ends or protruding ends, and as they are complementary to each other, they stick back again by means of this transient base pairing. So here the type of cuts, the staggered cuts, it is able to generate either 5' or 3' overhangs. Eco R1 is the enzyme which generates 5' overhangs. See here in the 5' end you are having single stranded DNA. So this is called as 5' overhang. Whereas PVU1, it is the restriction enzyme, it is producing this single stranded regions in the 3' end. So it is called as 3' overhangs. PVU1, it generates DNA fragments having 3' overhangs, whereas ECO R1, it generates DNA fragments having 5' overhangs. Coming to the blunt end, so here that is the enzyme is cutting exactly at the same position in both the strands, so you are getting fleshy ends or blunt ended DNA fragment. And let's see the applications of these restriction enzymes. We have known that the restriction enzymes, they are used mainly for isolating the desired gene from the source and also to cut the vector DNA. To insert the desired gene in the vector means first you have to cut the vector DNA. For this purpose in gene cloning they are extensively used, and also another application is it is used in RFLP analysis. RFLP analysis means restriction fragment length polymorphism. This particular technique is used for identifying individuals or strains of a particular species. Suppose any new species is identified, that one how it is different from that of the existing species, it can be known by means of this restriction fragment length polymorphism. Then it is also used in DNA sequencing experiments and also in the construction of genomic and cDNA libraries. In this manner, these restriction enzymes, they play a very key role in most of the genetic engineering experiments. Okay, with this I conclude the session. Hope you enjoyed learning this session. Last but not the least, I profusely thank Commissionerate of Collegiate Education for providing this LMS platform to reach a large number of enthusiastic learners like you. Thank you one and all.